Welcome and thank you for joining us for today's webcast, Emerging Trends in Bioabsorbable Vascular Scaffolds, sponsored by Zeus and TechBriefs Media Group. I'm Billy Hurley, Associate Editor with TechBriefs Media Group, and I'll be your moderator today. Our webcast will last approximately 30 minutes, and there will be a question and answer period at the end of the presentation. If you have a question, you can submit it at any time during the presentation by entering it in the box at the bottom of your screen. Our presenters will answer as many questions as possible at the conclusion of the presentation. Those questions not addressed during the live event will be answered after the webcast. In order to view the presentation properly, please disable any pop-up blockers you may have on your browser. At this time, I'd like to introduce our speakers. Dr. Bruce Anot, Corporate Director of Research at Zeus, has extensive experience in bioabsorbable polymer synthesis and processing with numerous innovations in related devices and components. Dr. Anot leads a diverse team of scientists and engineers within Zeus's ISO 13485 accredited research facility and corporate analytical lab. Also on the line today is Dr. Jim Oberhauser, medical device consultant. Dr. Oberhauser received a PhD in chemical engineering from the University of California, Santa Barbara, and completed a postdoc at Caltech, focusing upon polymer rheology and strain-induced polymer crystallization. He led product development of Abbott Vascular's Bioabsorbable Vascular Scaffold from 2007 to 2015 and has made significant contributions to the development of this novel therapy for coronary artery disease. So now I'd like to hand the webcast over to our first speaker, Dr. Bruce Anot. Bruce?
Thank you, and thank you everybody for attending our webinar today. As you heard, the title of today's webinar is Emerging Trends in Bioabsorbable Vascular Scaffolds. But just to recap for those who were falling asleep last time, go through the standard treatments or the history of treatments for coronary artery disease. As you know, coronary artery disease happens with a narrowing of the coronary artery. In years past, angioplasty was the treatment of choice, where they would dilate a balloon within the plaque, pushing it into the walls of the artery. And then along came bare metal stents as they saw a need to help support the lumen of that artery, given some of the damage that was done during the angioplasty procedure. That was soon followed by a couple generations of drug-eluting stents that have helped reduce the proliferation of new tissue within the vessel and maintain greater patency. And now we're in the realm of bioabsorbable vascular scaffolds with a couple CE marked or close to being CE marked. And Zeus has been heavily involved in the development of biaxially oriented tubing that is lased into bioabsorbable vascular scaffolds. So the evolution of strut thickness, as you can see with the first generation of drug-eluting stents, the cipher started out somewhere around 140 microns in wall thickness, followed by a couple others, and slowly started to go down below 100 microns. After several studies that showed a direct correlation between the strut thickness and changes in blood flow at the vascular wall, that was leading to thrombosis and reocclusion of stents. They went to thinner and thinner walls, down some down close to 70 microns, most of them in the 80 to 100 micron range. And then the first generation bioabsorbable vascular scaffolds came out with a strut thickness close to 150 microns, and there has been a recurring movement of trying to get those stent struts thinner and thinner, and I'll touch on that some more. Zeus currently has an oriented tubing for bioabsorbable vascular scaffolds that is right at 100 microns. So what is needed for a clinically successful bioabsorbable vascular scaffold? You need acceptable shelf life of the product, the materials in the products, and this is a combination you can see here of the materials, packaging, and sterilization, followed by ease of deployment. Again, somewhat mediated by the materials, certainly the design of the stent and the design and componentry of the delivery system. Successful deployment with some overexpansion. This has been one of the limiting factors with the 100% PLA stents to date, is the inability to overexpand them to a level that practitioners are accustomed to. sufficient radial strength during remodeling to help open up or maintain patency in the coronary artery, and low recoil to maintain that vessel patency so that you don't get some late loss of the luminal diameter as the scaffold starts to collapse down. Strut thickness that limits turbulence, and I spoke to this with the thinner stents or walls that we're making in our tubing, and we'll talk more about that later. The absorption profile that complements the rate of remodeling, this is really dictated by the chemistry and material chosen to make the tubing for the stent or to make the stent out of, and we'll touch on some of that in just a minute. And then biocompatibility with a low incidence of hyperplasia, which is a combination of the material, certainly to some extent the design with the lower strut thickness that I've mentioned, and the drug coating that is going on these stents. So when designing tubing for bioabsorbable vascular scaffolds, there are really three major components that are pulled in, and that's a combination of the polymer chemistry, the polymer processing, and the geometry. So to start with, on the polymer chemistry, you select polymer chemistry that is conducive to the healing time with bioabsorbables to the tissue bed in which it's being implanted. For bioabsorbable vascular scaffolds, this is started with one of the longest-lasting standard bioabsorbable polyesters, poly-L lactide. And as you can see in the bottom left graphic there, those pendant groups or side groups on the PLA decrease the availability of the sensitive linkage in the backbone of the polymer chain to water. So it is a slower absorbing polymer compared with PGA, which does not have those side groups. You can see that on the bottom right there. So material selection for the homopolymer PLA has been chosen because it was felt that there was a very long duration of support that would be needed before the scaffold started to lose its mechanical integrity and ultimately be absorbed. We're learning more as we see more and more of these being used that some shorter durations or faster absorption times may be preferable. So when you combine the polymer chemistry with the polymer processing, which is critical for coming up with the final polymer morphology that really dictates what you can do with the polymer tubing and the properties of the stent once lased. So in order for the polymer morphology for the bioabsorbable vascular scaffold to be right, you need to be able to set it up so that you can achieve three different primary properties there. The mechanical properties in terms of the material itself, the dimensional stability, once it's crimped, then once it's deployed, that it will maintain those dimensions and you get very low recoil so that you don't lose your luminal diameter there. And then the final is the absorption profile. And so the polymer morphology, which arises from the chemistry and the processing together, is very important for these critical performance factors. Going further with the polymer morphology for the bioabsorbable vascular scaffolds, careful control is needed to ensure proper orientation and crystal formation during the processing and deployment of the stent. The polymer chemistry controls the crystallinity. For the most part, you can have both amorphous polymers and semicrystalline polymers. PLA certainly falls into that semicrystalline bucket. And then the polymer processing dictates or helps control the crystallinity as well as the ability to achieve proper molecular orientation. Because without the proper molecular orientation and establishment of proper crystal size and morphology, you will not have the mechanical properties needed for a polymeric stent to function as a metallic stent. So for the polymer morphology, it's important to achieve high crystallinity with biaxial orientation. And the polymer morphology dictates the ability to do this biaxial orientation and give you proper strength in both the radial and the axial direction. And this is tunable in terms of where most of the orientation goes for property balance and for particular stent designs. This also provides increased toughness and flexure modulus and thermal and dimensional stability for the tubing as well as the glazed scaffold. So in producing a clinically successful bioabsorbable vascular scaffold, combining the morphology and geometry, when combined effectively, will definitely yield a clinically successful BVS. So we're starting to understand more about the geometry and how to set up the proper polymer morphology for the subsequent processing steps that dictate the ultimate properties in the final scaffold. So you need to have exacted control of the polymer morphology in the tubing that will be lased, and you need to balance the input tubing mechanical properties with that of the final scaffold design and geometry to make sure you achieve optimal orientation. So in terms of progression of oriented tubing over the years at Zeus, control of the polymer morphology, exacted control of that polymer morphology has enabled us to improve our biaxial orientation to create stronger and tougher tubing with thinner walls, which ultimately translates into thinner struts. And you can see there on the far left our first generation 150 micron extruded tubing for bioabsorbable vascular scaffolds, and some of our latest work moving towards the 100 micron wall, where we actually are able to achieve a higher strength in the 100 micron wall tubing than we were able to in that early generation 150 micron wall tubing. So we keep talking about strut thickness and the importance of strut thickness and where that goes. There have been multiple clinical and modeling studies that have shown a direct and independent correlation between strut thickness and restenosis. With this, there are many mechanical and biochemical factors that contribute to the low endothelial shear and the disruption of normal vasomotion, increased arterial wall damage, and decreased protective biochemical production with the thicker strut stents or thicker strut bioabsorbable vascular scaffolds. And this is done through a number of ways, but it's a coupling of the mechanical damage during the deployment, the amount of material, as well as the low endothelial shear stress that promotes platelet aggregation with the thicker struts when you have that low flow on the back side of that strut. So now as we go from oriented tubing to the bioabsorbable vascular scaffolds, we've talked about the proper material chemistry, processing, and control of the polymer morphology are all part of the critical first steps, at least in terms of creating the biaxial oriented tubing that Zeus produces. But all the subsequent steps are critical as well, whether it be the handling, the actual laser cutting, the drug coating, the crimping process, the crimping temperatures, and the technique used for the terminal sterilization. And it's critically important to consider all of these steps during the production of the oriented tubing for a bioabsorbable vascular scaffold. And so I'm going to turn it over now to Dr. Jim Oberhauser, who's going to discuss some of the work that he's done in terms of watching some of the different effects from the crimping and deployment of bioabsorbable vascular scaffolds and the importance for communicating that to achieve the ideal input tubing possible. Jim?
Thanks, Bruce. So Bruce spoke a lot about the polymer morphology, so I'm going to delve into that in considerably greater detail and try to hammer home the message that, starting with tubing is certainly a very important first step, but the decision making you use in selecting the tubing morphology is critical to also consider those subsequent manufacturing steps. So I'm going to be talking about work that I did in collaboration with Julie Kornfield's group at Caltech in chemical engineering. And this is the basic principle of what a BVS looks like. I'm going to focus primarily on the PLLA backbone, which, as Bruce alluded to, PLLA is a semi-crystalline material. It's the material that is central to delivering strength in the device, and so we want to talk about how we deliver that strength. And again, the point is that while these are materials that are simple to manufacture at its face value, but it's what's going on at that microstructural level that is everything in terms of the ultimate performance. So again, this is a repeat of what Bruce had mentioned before. You can have polymers fall into a couple of categories, purely amorphous or semi-crystalline, which would be a mixture of crystalline and amorphous domains. And it isn't necessarily strictly the quantity of crystallinity so much as it is the quantity of crystallinity and the orientation of those crystallites that governs the macroscopic material properties. So things related to hardness, ductility, stiffness, strength all governed by how much crystallinity you have and how it is oriented relative to the geometry and the deformations that are being posed upon the device. So I want to take just a thought experiment here. We'll start with the tubing as a larger diameter tube, and this is typically one might start with. Then you laser cut a pattern into it. And then, of course, the next two processes are also deformation processes. We take this initial diameter of this laser cut tube and crimp it down onto a balloon dilatation catheter. These are approximately 50% strains at some of these junctions, these hinges as you might call them. And then we do the exact reverse process to go to deployment. So the amount of stress that's being applied to these hinge points is tremendous and in many ways paradoxical in the sense that we want the material to respond very favorably to crimping and to go with it. But then again, post-deployment, you want it to resist the same deformations that are going on in crimping. So it is a paradox. And you're also doing it with, in this case, a pure PLLA system, which is a notoriously brittle material. And while Bruce outlined the limitations of post-dilatation for some of these devices, in many ways what's going on with these materials is remarkable. The deformations that they're absorbing and the ability to be able to post-dilate at all is in many ways an accomplishment. I want to go through some of the material level questions of that kind of explain how we get to that point, how it's so successful. So this is just a schematic of traditional blow molding. It's not necessarily identical to what one does in forming tubes, but it's emblematic, again, of stretch blow molding is the same sort of philosophy of you're blending in different radial and axial strains in this process. It's the strain-induced crystallization process that is driving the initial orientation and number count of crystallites. So you're setting that initial polymer morphology through these high-strain processes that are occurring above the material's glass transition temperature. And if you look at these tubes where you're shining X-rays, passing X-rays through what I'm going to call the radial direction of the tube, you can see these very strong reflections, these equatorial 110, 200, and as well as these 203 reflections, all of which are emblematic of the fact that you've got strong orientation of the main chain axis around the circumference. So if you're trying to build up high hoop strength, this is the kind of orientation state that you would draw up. Now, we're not able to, in this particular test, look at variations along that radial direction. We'll come to that topic here momentarily. But this might be an initial state that one would be aspiring to. If we zoom in on this lathe scaffold and then contrast this hinge point, if you will, in the crimped scaffold, where I'm referring to IB as an inner bend and OB as an outer bend, that's terminology that I'm going to use, you can see that in going from the lathe scaffold to the crimped scaffold, there's some substantial deformations. Along the inner bend, where the radial direction, again, is coming in and out of the plane of this picture, you can see that there's bulging along the inner bend. And in fact, along the outer bend, there's actually a bit of a reduction in the strut thickness in that area. So you're subjecting the material in crimping to massive compressive stresses along the inner bend, and actually then substantial tensile stresses along the outer bend. Now, these are all occurring typically below the glass transition temperature, and so one might think, of course, that, well, this morphological state is frozen in, and therefore I must not be changing it. But in fact, that's not actually the case. So if we begin first where we're going to take some slices of this particular hinge point, and we're beginning where 0 microns is the bottom, approximately 150 to 165 is the top, we're taking 15 micron slices through the system. And initially we're looking at this under polarizing optical microscopy. And what you see basically is if you're looking at the top edge of things, there's very little color contrast, which tells you that the degree of orientation state in that system is nominal at best. There's very little going on. On the other hand, if you move closer to the bottom surface, the colors of the Michel-Levy colors become much richer, much more colorful, and they're much more oriented. So for example, in the C45 diagram, you can see the bright band of orange along that outer bend, which had been placed in tension during the crimping process. What you're seeing there is a high level of birefringence, high degree of orientation, high degree of crystallinity. That's completely different than was observed in the upper layers. And so what you're seeing is that during the crimping process, and perhaps as well during the original radial expansion process to create the tube, there were probably morphological differences along that radial direction through the system. crimping is now exaggerating that, so tension is driving crystallinity along the outer bend, and then along the inner bend you're seeing substantial crazing that's going on. So that was where the bulging was, around the inner bend. And so the material is adapting to these substantial stresses. We're going to now apply X-ray microdiffraction to these same systems, and I'm going to take that same C45 example here momentarily, and what I'm going to do is I'm going to take 200 nanometer diameter hole areas, and I'm going to sample them in 5 micron steps from the inner bend to the outer bend. And we're going to look at what those X-ray patterns reveal there that support what we think we're seeing in the Michel-Levy polarized optical microscopy. So if we look at this, again, 25 microns is clear near the inner bend, 160 microns is near the outer bend. And what you want to see is that at the inner bend, there's this relatively disordered crystalline state. There's no sharp peaks in the X-ray scattering patterns until you start getting to around 55 microns in. There you're starting to resolve a bit more of orientation. It's where the orange colors start to build up in the Michel-Levy diagrams. And as you move from 70 to 85 and so on and so forth to all the way towards the outer bend, the crispness and the width of those azimuthal scattering patterns is becoming tighter and tighter. So you're getting a stronger and stronger sense of orientation of these materials. So while you look at this initially and you think that you're tubing and you're crimping, that you have this nice homogeneous morphology, at this stage of the game it's anything but. By contrast, now what we're going to do is we're going to take that same crimped scaffold, now we're going to, well, not the same one, but a different one, and now we're going to deploy it in 37 degrees C water, and again continue to assess how does the morphology evolve through that process. This is mimicking deployment in vivo. And first thing you want to make note of is along that inner bend, where we had seen substantial crazing before, one now sees these diamond-shaped voids. These are observations that one had been made in hinged polymers dating back as early as the 1970s. So it's a very common feature for stress relief in systems that have been subjected to substantial compression. Now, if we look at the polarizing optical micrographs, where again the lower numbers correspond to the lower portion of the strut and the higher numbers correspond to the top of the strut, you see sort of a similar example at the very top, but beginning at D106 all the way down to D13, there's much more homogeneity of orientation. Those orange Michel-Levy colors are more ubiquitous throughout the whole thickness of the sample. And moreover, particularly as you get deeper into the systems, if you move along the inner bend to the outer bend, those colors are again more consistent. So deployment also appears to be having an effect upon morphology. We'll do the same microdiffraction experiment. And here we're going to make a comparison, again, moving from the inner bend to the outer bend. And what's striking is now all of a sudden the material is almost entirely uniform and oriented around that hinge point. So this is an astonishing transformation of a material in really what is a sub-TG deformation. And this strong orientation that's being delivered directly impacts the final mechanical properties and performance characteristics of the device. So again, contrasting the two states, the crimped scaffold and the deployed scaffold, and particularly want to draw your attention to those X-ray scattering patterns, you can see again the consistency of the deployed scaffold, whereas the crimped scaffold is still in this evolutionary state of multiple morphologies. So I just want to conclude with a few points here. So obviously this marketplace for bioresorbable scaffolds is being launched into an existing $4 billion category. Interest in these BVS products is of course increasing, and there's large trials that are underway to demonstrate safety and effectiveness. Ease of use remains the major obstacle, as Bruce alluded to, so strut thickness and the ability to crimp down to lower profiles while still maintaining the performance characteristics of a traditional metallic drug-eluting stent are fueling all sorts of innovation in the area. But the key limiting step to achieving this is a detailed polymer microstructure and the management of that microstructure throughout the totality of the manufacturing process. So you have to consider every deformation manufacturing unit operation as having an effect on microstructure and not just the initial blow molding step. So this is the example of working with a company like Zeus you want to work in partnership so that you have full visibility, both sides, along the whole manufacturing path, so that if they're going to deliver tubing to you, they have visibility into what your ultimate objective is. So you have to appreciate the interaction of microstructure and manufacturing process deformations. That's the central key to overcoming these devils in the details. So without any further ado, I'm going to turn it back to Billy, and we'll treat any questions.
Thanks, Jim. At this time, we'd like to begin our Q&A, so if you have a question, you may submit it by entering it in the box at the bottom of your screen. We have time for a couple questions. The first question we have here, does Zeus measure the shelf life of its PLLA tubes?
Yes, we do. This is Bruce. We do measure the shelf life of all the bioabsorbable materials that we process and we make here. We have ongoing shelf life studies for everything ranging from polyglycolide on up to the polyolactide and different copolymers and terpolymers in between.
Here's another question. How does your material compare to metallic stents?
Well, we don't actually make any finished stents, so it's kind of hard to compare our material directly to metallic stents. But we have worked with customers who have obviously lased our tubes into bioabsorbable vascular scaffolds. And in terms of radial compression force and some other mechanical properties, we, in a lot of instances, have mechanical properties that are comparable to, if not better than, some of the top-running permanent metallic stents.
So Bruce, and I can add to that actually, since I have done comparisons, and the answer is that it strongly depends on those downstream manufacturing processes after you've formed the tube. I've done it in such a way that you can be very successful and achieve comparability even with 100 micron strut thickness, and I've done it in such a ways where those downstream manufacturing processes have been an unmitigated disaster, and I have a horrific result. So the beauty of the answer is it depends, and you have to consider all of these manufacturing steps in confluence.
All right, we'll end it there. That concludes today's webcast. Again, if we did not get a chance to answer your question today, our sponsors will do their best to address them after today's presentation. So our thanks to Dr. Bruce Ano, Dr. Jim Oberhauser, and everyone out there for joining us. And just a reminder, this webcast will be available on demand at www.techbriefs.com for the next 12 months. Have a great day.